Well, thank you all for coming, and wanted to tell you a little bit first before I start what Trovagene is. Trovagene is a publicly traded company. It's actually it's a restart, and I won't go into the history of it, but it's a company that started actually almost 10 years ago with the scientific founder being a Russian scientist trying to solve the problem of monitoring radiation toxicity from Chernobyl. So this was back quite a while ago when there was that meltdown, and so that's really where this company really started. I joined the company about a year ago, but about 2.5 years, 3 years ago, there was a large management change, and Tony Hsu became the CEO of the company, and they took it public, and we've really focused Trovagene now as a molecular diagnostic company, and with a primary focus in metastatic cancer patients, monitoring metastatic cancer patients by the use of cell-free DNA, which I'll be talking to you about today. So that's kind of gives you a little bit of a background of where we are and where we've come from. And the other thing I was going to state is that Trovagene is really let's just put it this way, our goal is to really become a clinical company, a company focused on the clinical aspects and being relatively agnostic to technology and using whatever technology we need in order to accomplish that goal. So I will talk about a couple different kinds of technology as I go through this. I think that, of course, this is very myopic in my view here, but I've put cell-free diagnostics right in the middle of this slide, this is to try to make a point of where we think, and not only ourselves, but a lot of the leaders in the field now in molecular diagnostics think that cell-free diagnostics is coming of age and will play a very important role in patient care going forward in oncology. I think we all know that there is a large body of knowledge that has been developed, and we now know that tumors are driven by oncogene mutations. The last two talks really demonstrated both at the single cell level as well as some of the other discussions in the last talk regarding how heterogeneous the tumors are today. And this has resulted in, the effectiveness requires monitoring the entire complexity. Resistance, we know, develops through known molecular mechanisms. For example, in lung cancer, if you have EGFR mutation and you receive Tarceva, then 50 to 60% of those patients will develop another mutation in EGFR of the T790M, which then is now being targeted by at least two companies, Clovis and AstraZeneca. And so the other thing is that the standard of care today for monitoring metastatic cancer patients, of course, is imaging. There is no molecular information that's given to that. We also know that tissue biopsies, of course, are also standard of care, but in the metastatic setting, this, of course, is difficult. It can have morbidities associated with it, and also back to this whole problem of tumor heterogeneity. And without being too provocative on the first speaker talking about CTCs, it's clearly the enumeration is something that's clearly been demonstrated with CTCs and the use of enumeration as a prognostic for prostate, breast, and colorectal cancer. But only maybe through single cell would you be able to reach this heterogeneity issue that is one of the problems associated with CTCs. I think that one of the things that I would say is that the reason cell-free diagnostics or cell-free DNA is really becoming of age is really because of those three pillars I've kind of brushed over. Number one is now we have technology. We have NGS, droplet digital PCR. We have ways now to measure and find that needle in the haystack of 10,000 wild-type genomes in one mutation or several mutation fragments that are associated with the tumor. That before did not exist. We didn't have that kind of technology several years ago. The other thing that we have today is we have basically, we now know that because of tumor heterogeneity, that this is really might become much more important. And then the third thing is that we know that tumors are continually changing, as has been brought up by both speakers before me, that tumors change over time. Basically, like for instance, the EGFR story with the T790M, the resistance. And so needing to monitor is becoming much more important in basically patient care, particularly in stage 4 disease. So I think that that's really the argument that I make and put forward to you. And the next slide is really telling you a little bit about cell-free DNA and what it really is. And if you focus over here on the left-hand side, this is a blood vessel, and basically you have a tumor there. And tumors, when they are growing and multiplying, dividing, or also when they are being killed by a particular therapy, they will die and they will release their genomes into the bloodstream. Now, that means that there's basically naked DNA floating around in the bloodstream that could be, i.e. plasma, via plasma. One could, in essence, monitor or measure the number of fragments found that are floating in that bloodstream of that patient. Now, I think we all know that once the bloodstream goes to the kidney, and then basically urine is basically the end product, the waste product of that. And so urine also offers another viable specimen for basically measuring these fragments. And that's what I will talk to you today about. I'll talk to you mostly about urine, but I also will show you some plasma data as well. So just to kind of go over this, the fragments of DNA from tumor cells that have undergone cell death through apoptosis or necrosis are excreted through the bloodstream to the urine. This can be molecularly measured, such as like with EGFR mutations. Tumor heterogeneity indicates that a single biopsy may not encompass the entire story for the patient. And it's been shown by numerous investigators over the last 4 or 5 years and published in high-profile journals that tumor load of the patient, so if measuring the actual number of tumor, of mutation fragments, let's say, for example, we're looking at KRAS, and we're looking at a particular mutation. If we actually count the number of times we actually see it in the plasma or in the urine, this is associated with the tumor load, or proportional to the tumor load of the patient. Even more importantly, it is correlated with responsiveness to therapy. That is to say, if you have less number of fragments, if a patient has a high number of fragments of a mutation and it goes down, this correlates actually with the patient responding to the therapy. So this is a way of tracking responsiveness, and specifically, and the point I would make is that versus the some of the old CA-125 and some of these other markers, these are markers that are indicative of the actual tumor genome. And so this allows us to very directly measure the number of fragments present in the plasma or the urine. And then more importantly, it's the emergence of resistant mutations such as the T790M, where you're able to see those things. So let me go to the next slide and talk a little bit more about this. So what would be the clinical utility of this? How would a physician use this kind of information or this test? I mean, first of all, it's a way of very quickly detecting a mutation. Sometimes, and I'll show you in some patient populations of certain diseases, it's difficult to get a biopsy. If you can't get a biopsy, another way to do this is to look at cell-free. And also, it eliminates, the morbidity associated with it. Monitoring tumor load, you can have a faster detection of the responsiveness to therapy. One doesn't have to always wait for the next CAT scan or when they come back in. One can measure very quickly, is this patient responding to therapy? And then, of course, emergence of faster detection of resistant mutations, detecting the tumor heterogeneity. So these are kind of some of the high-level, at this point, proposals of how cell-free could be used. And one of the things that I'm looking forward to is that and it was mentioned by Jeff in the first one of his slides, is that Trovagene has now set up a collaborative effort with CHI. And so we're very interested in trying to take this technology and see if we can work with some of the physicians, some of the medical oncologists within the CHI organization to see how can we utilize this kind of testing. And, you know, as I was talking to one of your board members earlier, Stephen Moore, basically, we don't want to just come here and try to do some kind of cool science. What we're really interested in is trying to build a relationship with CHI such that we can also identify how can this save money for the organization and at the same time benefit patients. And so we are very much looking forward to that kind of dialogue with CHI going forward. Now, I think that a lot of people, we talk about the tumor genome, and I think that there's always been two different camps. There's the camp of, well, it doesn't really matter what kind of tumor it is. If it has a BRAF mutation, you treat it with verapamil or a BRAF inhibitor. Now, we know that that's not actually the case anymore. We know that, for example, in melanoma, that 50-60% of patients have BRAF mutation, and we know that there's a very high responsive rate to a BRAF inhibitor in melanoma. However, we know that also that the trial had failed in colorectal cancer. Patients who have a BRAF mutation in colorectal cancer do not respond to a BRAF inhibitor. So there is some kind of cellular contextual information that is also important beyond just the actual tumor genetics of a particular tumor. So the approach that we have really been taking is really one where we are looking at cell-free genomic mutations and alterations, but also keeping in mind that these are very different kinds of cancers, and so that different kinds of mutations mean different things in different cancer types. I'm sorry if I stated the obvious. One of the things I want to talk about, just one of the things about the practical advantages of urine, although Trovagene is really a company of cell-free molecular diagnostics, we got into this by urine-based testing. But we also recognize that really to the physician, what we're really talking about is what's the easiest and most practical way that a physician can we can help them monitor his or her patient. So clearly, I pointed out a couple things. High-frequency testing for longitudinal tumor dynamics. What I'm really talking about here is the fact that one can do home sampling, I guess you might want to call it, where a patient can just basically pee in a cup or a vessel, and then it can be sent for testing. It's very simple. There's no blood draw. Nothing else has to be done. So this can become important when you want to look at, let's say, weekly urine samples to see how a patient's responding to therapy. Something that's not as applicable or practical, I guess I should say, for a patient have to come in or have somebody come out and do a blood draw every week. The other thing is the ease of sample collection is obviously truly non-invasive, no physician or healthcare provider needed. There's also the fact that the volume, of course it does depend, if you've already peed, then you probably won't have very much urine to pee again. So there is no volume in a sense a limitation. Sample processing stability, there's no need to centrifuge or extract prior to shipping. You can ship at ambient temperature, it's stable. So these are practical things, but they actually, we believe at Trovagene that in talking to some of the physicians that we work with already and collaborate with, that this actually becomes important in day-to-day life of stage 4 patients. And also, I would argue that this is also a systematic representation of the total tumor heterogeneity. Of course, urine and plasma are both are doing that. So what is Trovagene? I said Trovagene was agnostic to technology, but of course what we have done is we've built what we would call in the industry, we've built a proprietary platform, which really what that really means in language is that we have basically developed methodologies and technologies in order for us to be able to take a urine sample, in this case, and be able to get the DNA out of that in a way that it's purified, and from that allows us to then be able to analyze that urine sample. Now, one thing that I will state that, although I've already stated it, but I do this because of sometimes people think that we're a urine-based company, and so we're basically looking at prostate cancer. What we're looking at in the urine is actually DNA fragments that did come from the circulation. So we're measuring fragments from tumors, solid tumors, that have dumped their genomes into the blood, and then they're now being excreted into the urine. So that's what we're looking at, what we'd call systemic DNA in the urine. First of all, there's the extraction and isolation of small DNA fragments. So in plasma, the free DNA floating around in plasma has about an average size of about 150 bases, to get a little technical here. In urine, it's graded even smaller. It's down to maybe about 75 bases. So in other words, if you're, from a technology point of view, if you're going to actually interrogate this, you need to be able to work with very small, what we call amplicons, i.e. any of those people in the room here that do PCR, you understand what I'm talking about, that you have to build your assays to very short amplicons to be able to measure these different mutational changes. So we've worked on that quite a bit at Trovagene. And then I think the next thing that you have to think about is that in tissue, if you're talking to a pathologist, an anatomical pathologist, and you say, well, I'm going to run the KRAS test on this patient's biopsy, the pathologist will say, well, I have this kit and it has a 1% sensitivity. That is to say that I can measure a KRAS mutation in one tumor cell out of 100 that don't have it. And we know that that is probably the bottom end for clinical applicability as well. A good example would be EGFR mutations. Now, if you're talking about plasma, what you're talking about now is a lot of other cells are dumping their genomes into the bloodstream as well. So a 1% sensitivity no longer really actually helps you. It's not sensitive enough. And so really you have to be probably more like 2 orders of magnitude more sensitive. So you have to be more in the area of 0.01%, or basically being able to see 1 out of every 10,000. So this requires also another technical challenge for those people who are in both plasma and urine cell-free diagnostics. So we have to do what they call an enrichment. We have to somehow enrich. Somehow we have to either at this stage or at the bioinformatics stage or both, we have to come up with a solution that allows us to have the sensitivity to be able to see these mutations in these patients. So we basically have worked that out at Trovagene, and we've come up with a method that allows us to get those 2 orders of magnitude that I was just talking about. So it allows us to be able to interrogate plasma and urine from these metastatic cancer patients. Now, like I said, the actual platform where we measure it, we're agnostic to it. I'll show you data mostly from RainDance. RainDance is a technology that allows you to do what they call droplet digital PCR, which in essence, for those of you that don't know what that is, what that really is that you do a normal amplification, but now every single molecule, every molecule is in its own little bubble or droplet, and so that allows you every reaction to be an individual reaction. This becomes, so you can have, RainDance has 10 million droplets, so you can do 10 million individual reactions and then put them all back together again and measure it. And so that becomes important because technically what happens if you treat everything as a population of DNA, sometimes those very rare events can't be found. But if you actually isolate them as individual entities in little bubbles of their own world, you can actually, it has a chance to amplify and for you to be able to see it. So we do that as well as show you some technology. We also are using the Illumina technology, the so-called next-gen sequencing or the massively parallel sequencing analysis. And we are moving a lot of our assays to the NGS platform simply because it allows us to do multiplexing, allows us to look at multiple genes at the same time, and also we think it has much better quantitation. I won't get into more of those details, but that's really where we're moving in Trovagene in this field. And then once you, of course, the biggest part is the analysis of the data afterwards and being able to make that call. Does Mrs. Jones have a KRAS mutation or does she not? And so really being able to make that call and using different kinds of informatics and statistics to be able to say what kind of significance that call is made on. So this gives you kind of an example. This is just showing you, it wasn't really meant to get into the details, but you can basically spike in DNA in an assay, a number of copies, and look to see what you can see. What you're showing you here is in an NGS platform and one of our KRAS assay that looks at exon 12, 13, 7 of the most common KRAS mutations, that we can have a reasonably good increase. As we increase the number of input, we get a greater amount of output frequency. And what we've shown by doing multiple experiments over and over again, that we can reliably detect 7 copies in a patient's urine or plasma, which is actually a pretty small number of fragments floating around. And I think I covered this already, but this is really the challenge, of course, is low signal to noise ratio, i.e. you've got 1 copy in 10,000, or you want to get down into that range of being able to look at seeing things. And really it's all about down here at the down end stream is one thing I haven't covered, and I know some of the people in the room know this very well, that in NGS or in any kind of amplification method, you actually introduce errors into the process. And so these are artificial errors either by the enzymes you're using. They have their own error rate of copying things, so they make mistakes themselves. Or in the actual sequencing, there can be mistakes made as far as the actual sequence. So these are artificial errors that can actually tell you that erroneously that the patient has a mutation or has a wild type. So there's a lot of work that goes into making sure that we can eliminate those errors, and then, of course, that becomes the noise at the level where you can then start to detect things. Well, in diagnostics, there really is always the question of your roadmap of clinical evidence. And when you're talking about any kind of molecular diagnostic, those of you in the room that are in this field know that this is always a road to the clinical evidence, the pillars of evidence that one has to develop or demonstrate through clinical studies is actually can be an enormous road. Now, what we're talking about here are biomarkers that are already clinically validated. We already know that EGFR mutations, those patients respond, it's part of the guidelines. However, we still have to demonstrate that cell-free DNA is clinically validated and can be useful in the practical setting. So we still have a road to go. And the first stage is really just purely diagnostic, really demonstrating that the mutational status of a patient, let's say BRAF, in the urine or in the plasma is concordant with the tissue biopsy of that patient. So when you're first starting out in this area, you have to basically show that, well, this patient, we already know they have a BRAF mutation because of a tissue biopsy. Can you actually see that in the plasma or in the urine? Now, in actual practicality, one would think that that's just your first step and there's really no clinical utility there, but I will show you a certain disease where actually getting a biopsy is so difficult and problematic that this right off the bat, if you can show that you have basically concordance, this already right away becomes clinically useful in the real world. Secondly, of course, is really quantitatively assessing and showing correlation of responsiveness to therapy. And I'll show you some of the data that we have on that as well. And then, of course, the third, in order to get adoption, really one has to do what we call an intervention trial, where you would actually use the information based on cell-free status, cell-free DNA diagnostics, use that information to change or treat the patient, and then show that that actually increases overall survival of that patient or that group of patients. So an intervention trial is your gold standard at the end for any molecular diagnostic. It's also the way you get paid. Now, let me tell you a little bit about some of the data, and this data actually, because it's a small audience, this is actually still confidential. It'll be coming out in ASCO, but I was able to show it to a small group of people outside of a conference. So this is really, it just gives you an example. This is some work that we've been collaborating with Philip Yanku at MD Anderson, and also a group of colleagues at Sloan Kettering. And this is a type of disease. It's a very, I didn't really know anything about this disease. Some of maybe the physicians in the audience have probably may have heard of it. It's called Erdheim-Chester disease, and also Langerhans cell histiocytosis. These are histiocytic diseases that were thought at one time to not be cancers or tumors. But several years ago, it was found that these patients, they're relatively rare, these are rare diseases, but it was found that these patients actually harbor a BRAF mutation, the V600E. And on top of that, then there was just a publication last year showing in 3 patients that they had dramatic responsiveness to BRAF inhibitors. And so that really changed the landscape, and of course, for these patients, it entirely changed their lives. And so the problem with this disease, though, it's a tissue-based disease of histiocytosis it's activated macrophages that go into either the muscle, or I should say the bone, or the heart, or the brain. Those are the major places it goes. It's very difficult to get a biopsy, particularly for those patients where it's infiltrated into the bone. And so this was it just so happened it was serendipitous, but Philip Yanku, who is a medical oncologist in the phase 1 group at MD Anderson, he happens to be an expert also in this particular disease, and he thought that this would be a great application of this technology right off the bat. So we got involved with him on that initially with a couple patients, and then we moved it into a much larger, relatively speaking, larger patient population with our collaboration both with he and Sloan Kettering. And what we looked at here was histiocytic patients. We basically got urine samples from each patient, and then we evaluated those patients which we called having the V600E mutation. We called those urine positive, and then we had negative. And within each of these groups of positive and negative, we evaluated the tissue biopsy, the BRAF V600E, for concordance. In these cases, in most of these cases, it was known. So we did know, we had to have a truth to be able to do this study to evaluate these cell-free technologies. And then we also looked at plasma. And then for those that were negative, we looked at concordance to the tissue, the gold standard in this case, and also plasma. Now in 22 patients, 18 Erdheim-Chester disease patients and 4 LCH. LCH, for those that don't know, mostly affects small children, ages 2 to 5 years old. It's actually a pretty, not a great disease to have. And so we were looking at both of these, though, because they're both considered to be histiocytic diseases. We had 22, and of that, 12 of them we called positive. This was a prospective, blinded study. We had a prior protocol. We did the study blinded. We sent the results back to both Sloan Kettering and MD Anderson with our results. They held the truth with the tissue biopsy. There was 10 that, of the 12, they actually had tissue biopsy on 10 of them. It was 100% concordant. In the plasma, which we actually ran as well, we had a 7 out of 8 concordance for positive. In the 10 urine negatives, of those, they had 6 with tissue biopsies, and they were 100% concordant. And in plasma, we were 8 out of 8 concordance. So we had a very, very good concordance of both the urine and plasma in concording it to the known, when they did have a biopsy of whether it was a BRAF V600E mutation or a wild type. And another study that we've been doing and ongoing and will also be published in ASCO is we've been looking at metastatic cancer patients. And here we've plugged into the phase 1 group at MD Anderson with Philip Yanku and his colleagues. And the objective here was really to measure the degree of concordance between the DNA mutational analysis from urine samples and versus the mutational status from tumor biopsy tissue. There have been reports out there today in plasma from other investigators that concordance is between about 75 and 80%. And so we were wanting to see whether or not we could achieve that similar kind of sensitivity with metastatic. Now, these are all comers, all different kinds of cancer, metastatic cancer patients. Then the second thing we wanted to do was we wanted to follow these patients. We wanted to get what we call, longitudinal data, that is to say, serial urine samples over time, and see whether or not the mutational status, the number of fragments that we detected, was correlated with responsiveness to therapy. Now, the concordance was 80%. We had 20 patients initially in the original pilot study. This move is now up to 33 patients. We're getting similar kind of data in around the 80 percentile for concordance with the gold standard. And what we also did is we wanted to ask I think even more important question was is there a correlation between the longitudinal data that we were collecting on these patients, this is another blinded prospective study, and with clinical outcome for these patients. And so what we showed was that of 17 patients that were able to follow serially, we had 15 that were evaluable that we actually detected out of these 17 that we were able to actually follow. 15, so most of the 89% of the patients that had data that we could actually serially follow, we were actually able to detect BRAF V600E. And we looked at two different things here. We evaluated time to progression. So for example, in these 15 patients, we looked to see if where over time, whatever the period of time was that we were evaluating them, that could be anywhere from 3 months to over a year. When we evaluated them, if the number of fragments went up, that was an increase. If the number of fragments from baseline to the end went down, that would be a decrease in the amount of fragments detected. Did that correlate with any kind of clinical outcome, particularly time to progression for that patient? And what we found was that when the urine cfDNA BRAF decreased, that is to say the number of mutation fragments that we detected went down, the actual median to progression was 259 days with a pretty reasonable confidence interval. If the urine over time CFBF increased, then the median progression was drastically reduced down to 61 days and with a reasonable CI here as well, with a highly significant difference between these two patient populations. Also, we looked at the percent change with respect to RECIST 1.1, and we demonstrated that there was a correlation between the number of fragments we detected in patients and the percent change of RECIST for each patient that we measured. I just want to show you a little bit of some of the examples of some of these patients. For example, here, this was a patient, a metastatic non-small cell lung cancer patient. We detected a BRAF mutation fragments here that went dramatically up over time. This is, for those of you that are in the back of the room, this is January of 2012, and this is February of 2013. So we obviously only got 2 time points here, but in this particular one, but we showed that basically this patient had progressive disease, greater than 74%, RECIST 1.1, and you can see that also this is the amount of number of fragments detected also went up dramatically as well. Now, and here's another patient where we had a lot more time points, and here we had a patient that at baseline prior to receiving a BRAF inhibitor plus a chemotherapy, or no, EGFR antibody, sorry, we had a very high level of fragments detected. This patient went into stable disease, minus 24% in tumor regression, and we also, this was followed by these time points here. We started in January of 2013, and our last one was in January of this year. And you can see that it went down, and then once the patient started to progress again, this also started to climb as well. Here's another example of a patient, another type of example where they had a baseline, very high level of a BRAF V600E, and then they received a BRAF inhibitor and chemotherapy. They had a stable disease, and they have continued to have stable disease out here, even though we had I'm not exactly sure what happened in this particular part of the patient. I have to find out. But anyway, just demonstrating that this is correlating with the responsiveness to therapy. Another example where the patient did not receive any therapy. This was over a period of May 23rd of 2013 to June 13th of 2013, and they just shot way up, and this patient went into hospice. Here's just another couple more examples of some things that we're kind of running across when we start to measure these over time. This is just another example. This is a melanoma. This is an appendiceal cancer, same kinds of stories. Here's a glioblastoma. It's kind of an interesting story where it looks as though we actually did not detect any fragments with this patient. It was known that they were known by tissue biopsy to have a V600E mutation. However, when they went into stable disease, because we were monitoring this a little more closely every couple months, there seemed to be a spike here. And so we're actually exploring that a little bit more because we think, our hypothesis today is that perhaps because when tumor cells do die, of course, they release all their genomes. And so it may be that very early on, you actually see an increase in the number of fragments detected if they are responding. So we're exploring that right now with a prospective study systematically with multiple number of patients and looking at weekly time points, right, from baseline, and then once they get therapy, a BRAF inhibitor, and seeing whether or not there is a point of inflection going up, and that this could be indicative, very early data prior to them ever coming back in for a scan within the first couple weeks, knowing whether or not they're going to respond to therapy. And here's another example of a papillary thyroid carcinoma where we see a similar kind of thing when we measured over a much shorter period of time, a peak, and then it went down and was below detection. So I think that, in summary, I just wanted to say that what we're really showing is that these kinds of technologies with cell-free, and I've been showing you urine, I showed you actually some plasma and urine data, that we really are able to show a reasonably good concordance with tissue biopsies, and also that the longitudinal monitoring is correlating with therapeutic response, and that we think that we can, because really CF or cell-free DNA from urine is really the most truly non-invasive method for really detecting clinically actionable mutations, monitoring the responsiveness, monitoring the progression, and also tracking the emergence of resistant mutations. So, thank you, and I'll take any questions if you have any. No, it's actually even easier than that. Did everybody hear that question? How do we? It's actually even easier than that. Basically, we have a little vessel, a little plastic container, and then it has the only thing you have to do is after the patient has peed into the little receptacle, you pour some purple dyed EDTA that's with the kit into it. And that basically, as you know, is a chelator of divalent cations, and then that inhibits any kind of DNases from degrading the DNA. And that's basically all you do. And then you can ship that ambiently.
And then do you bank all of that I'm thinking about your longitudinal studies. Do you
Well,
bank all of that, or do you just take your set and bank that?
well, what we do is we don't do any spin downs or anything. When it comes in, we have a couple alternatives, and we've done all of them. One is we can just freeze them at minus 20 or minus 80, and then thaw them out and run them all at one time, or we can do it prospectively as we get them.
It's a lot of space.
Well, you know, we used to do 24-hour jugs, and now that takes up space. And you can have too much urine. But I think what we've done now is we've in the last couple months, the R&D group has come up with a way of taking a normal urination is probably between 100 and 200 mils. And so they can take that, and in one fell swoop, we take the whole volume, high volume extraction. And so we go right from that right down to a very, to basically 440 microliters of DNA in TE. So, it's a much better than it used to be. Yes, David?
So with regard to using urine as a specimen, is it just convenience that urine gives you, or is there more to it?
I don't know, because what I more than alluded to was that that I think that where we started at Trovagene was, urine is the most practical sample to get, and so it's the easiest sample to get from a patient. Having said that, what we are looking at is, because urine is reasonably easy to get from a patient, we can do more what we call high-frequency testing. And high-frequency testing may allow you to, as soon as you get a baseline, and as soon as you put them on a therapy, every week you can have them pee in a bottle, basically, and you can know within a couple weeks, we think, we're doing the prospective study now, so it's a hypothesis at this point, that we would be able to tell the physician whether that patient is responding to therapy right away. And as we all know, stage 4, time is of essence.
So is there any time, like morning urine, evening urine, or after?
Well, yeah, we've our protocol. We didn't do a huge study in that, so we went to we did a limited study, and so we went to what we call morning void. When a person gets up in the morning, the first pee. Having said that, in all of our protocols, nobody ever pays attention to that. It's basically, whenever the patient comes into the doctor, that's when you get the pee. So we get it all over the place. It's whenever.
And is there any correlation?
We haven't really, the only way we can really, measure that is we need to do that study. We haven't really, at this point, we've been opportunistic and basically just, getting what we can get when we can get it, with the argument being that that's probably real world. And so our studies have to be close to real world as possible.
How easy is the multiplexing? Sorry.
Yeah.
Simultaneously a major multiple mutations.
Yeah, so that's a great question. So from a technical point of view, you've got this DNA. It's short, and what you want to do is you want to maximize the use of that DNA. And so right now what we've done is in the lab, in the R&D group, it's really been going after KRAS and then now EGFR, but as single multiplexes or sets of primers to go after that. Of course, what we want to do is we want to do all of them at the same time. And then, for those of you that know this, in essence, what you end up doing is you put in all your primers and you do what we call a pre-amplification, and we amplify all of them together in one tube, and then you basically take that to sequencing. Yes, Victor?
With plasma, there is always a great challenge to avoid contamination by the genomic DNA from nucleated cells. That's why essentially experiments with serum and cell-free DNA from serum have given very, very ambiguous, let's put it that way, results. here you have the danger of contamination from genomic DNA of cells that have been washed out with urine.
Yes.
How do you avoid this danger, and how practical?
Yeah, so that's a great question. As Victor stated, in urine, you've got the whole urinary tract, so you're getting epithelial shedding of genomes of epithelial cells that line that. So you're getting that. Not only are you getting that, you're getting bacterial genomes. Basically urine is a waste, as we all know, and it has all kinds of different genomes in there. Some of them are not human genomes, and so I think that one of the things that we've noticed right away is that, this is not perfect, this is more of a practical approach, the DNA that comes systemically is short. The DNA comes from epithelial lining or bacterial is very large. And so what we've done is we've optimized our DNA extraction for short DNA. And this allows us to enrich for this. And then, of course, what we are interrogating for example, is a KRAS mutation. Bacterial genomes, of course, we won't see that unless it's a cancer in the urinary tract. People don't float around with KRAS mutations, and so unless they have cancer, and so it's basically we interrogate for those mutations, and those, of course, are coming systemically. Yes.
From a clinical perspective, two questions for you. When you actually collect the sample, do you require a clean catch or a sterile sample? And then what's the typical turnaround time?
Yeah, for the first question, no, there's no sterility needed or anything involved there. It's basically a urine sample, and we put EDTAs put in there, for preservation. And what was the second question? I'm sorry.
For the physicians, what's your turnaround time?
Oh, I'm sorry, turnaround time, yeah. It's really about 5 days. Yeah.